Welcome everybody to our last March IMAG MSM working group meeting. I expect there'll be people logging in for the next few minutes. As usual, I need to remind people that this meeting is being recorded, it's being live streamed, and that the recordings will be made available afterwards. Appreciate those of you who are here. We have two speakers lined up, and we'll be getting to that in just a minute. As usual, I want to remind you that Reinhardt and I are the co-leads of the working group at the moment. We have web administration from Jim Sluka and other kinds of help from Jim Sluka. And you all hear regularly from Bruce Shapiro, who is on a well-deserved holiday this week. I also want to remind you that we have a Slack channel, Twitter channel, the IMAG MSM wiki page, IMAG LinkedIn page, and we have a YouTube channel. These only work if people use them. So please help us populate that channel to make sure that our efforts are seen. In particular, the YouTube channel has recordings of all of the speakers, all of the great speakers that we've had. And I hope that people will help us make sure that those videos are recorded, because we know that there are many people in the working group who aren't able to make this slot. Our Asian colleagues, it's awkward. For some of our European colleagues, it's quite late. And therefore, making sure that the recordings of the talks are available is important. We're trying a bit of an experiment today, and usually these things take a little bit of time to develop. And that is, there was a request that we have an announcement section. And the idea here is that people can make a brief announcement, try to keep it under a minute. If you have a slide, make it one slide. You don't have to tell us in advance. You're welcome to speak up now. But if you would like to let us know in advance, we could include your slide in the deck so you don't have to screen share. And we can also include it in the official agenda for the meeting, which may get a little more PR because it'll reach out to the people who are not just physically present, but to all the working group members. So I'd like to call now as a question, does anybody have any announcements for today? Again, this is new, so if you don't, that's fine. But we had a request at the steering group that we have this option. So I'd like to make it available. And you just speak up if you have an announcement.
I have one, James.
I want to announce something.
Sorry, Jacob. Sorry, hold on. Let's let David go first, and then Jacob.
Okay, yeah, sorry, Jacob. Yeah, it's actually was in my slide deck for my talk, but it's a meeting that's coming up. And I could share screen here just to show you the, I can't share screen, I guess. Okay, let me try again. There we go. It's called Visualize Med, and it's about bringing, wait a second here. Can you see that?
Yes, that's great.
Okay. Yeah, modeling and simulation for therapy development. This is the second version of this now. The first one was in Minneapolis a year and a half ago. This will be virtual, and encourage you to check it out. I'll be speaking in it and also organizing a couple sessions. Stacy Finley, John Burke, Allison Klaas. Stacy's at USC. John's the CEO of Applied Biomath in Cambridge, and Allison's in the modeling group at Novartis in Cambridge. And then there'll be a road mapping opportunity the day after to kind of scope out how can we get mathematical modeling more integrated into pharma and biotech industry and affecting FDA, improving FDA applications. So thanks.
That's great. I wish I'd known about it. That I missed that, and it's something that I think a lot of the working group members would find very interesting. So if you send us that link, we'll send that on to everybody in the working group as well. Thank you.
I put it in the chat.
Okay. Reinhardt, can you grab that and pass it on to to Bruce to send on?
Yes, we'll do.
Thank you. Jacob, you had an announcement.
Yeah, it's a pre-announcement. It's something that Sharif, Rahman Sharif, he's a little bit late, but he will announce it. But he's planning a workshop on teaching reproducibility. It's probably will happen around June. Look out for the announcements. I'm trying to let people know ahead of time, because if they have models, we will teach you how to do things properly and make it more reproducible. So start preparing your models. This is why it's a pre-announcement. Roman Sharif will be here. He'll talk about it more.
Okay, wonderful. Any other announcements?
Jacob, do you have a URL or something that we could use?
This is why I said it's a pre-announcement. Sharif is handling this. He's just a little bit late, so I didn't want to miss the opportunity. We need the people to start preparing their models. This is why I'm announcing it.
He's on the call.
He's here?
I just joined when you started to speak. Yeah.
So announce the workshop.
I think it's okay. You already did. So we will have a 2 days workshop. I think we did it last time as well. So I'm still finalizing the details. Once I have them, hopefully in the next week or so, I will share with everyone. So it's a hands-on workshop where everyone comes with their own model, and then we try to encode them in SBML or different tools and make them more reproducible and share with the rest of the community, and see if we can integrate some of these models together.
Okay, lovely. Thank you. That's perfect. This is exactly the kind of thing that we're hoping to have announced. I should say, I guess in the spirit of a pre-announcement, is that the last week of July, we will have our annual Tellurium Network Dynamics Modeling course taught by Herbert Sauer of the University of Washington. And then we'll have our annual CompuCell workshop the first week of August. And I hope by next week or the week after to have the formal announcements for that too. Wonderful. Well, thank you very much, everyone, for sharing your opportunities. And now I will move on. I just wanted to mention quickly that people last week, Reed from NIAID shared this NIH set of RFAs, which are primarily in data science, but may have some modeling component. I've written to the program director for clarification, and besides the statement that they're primarily interested in data science, I've not had a formal clarification yet. If I find out any more, I will report it. I know many people in the community might be interested in those, so I just wanted to make sure those were highlighted again. In the slide deck, there's the specific RFA calls. I'm not going to go through those in great detail today. Something that we are interested in that came up in the steering group meeting last week was that if there are new collaborations that have developed, and I know there are several, I know it's work to report those, but if you could report back to us on whether these meetings, either the direct meetings, the subgroup meetings, or the email lists have helped you build new collaborations, that is considered an important metric of the success of the working group. And so if you have written a paper with someone else in the working group, if you're writing a grant with someone else in the working group, if you're preparing a teaching and modeling resource, or even if you're simply bouncing ideas off people you wouldn't have met otherwise, if you could document that even in a sentence or two and send it on to us as the organizers, that would be extremely helpful for us in our planning and our working with the IMAG MSM team. Our upcoming meetings next week, we have 2 meetings on Thursday. I hope that is manageable for people. We have a special meeting with a longer talk by Rita Colwell, University of Maryland, who's going to be talking about her modeling and experimental work in epidemiology, focusing on cholera in particular. People will recognize her name as a longtime director of the National Science Foundation. and we're very pleased she's going to speak to us. And then in the afternoon, we'll have John Yin from the University of Wisconsin-Madison speaking at the regular time. And we're going to try the experiment of reserving a slot for group discussion, as was also requested by the steering committee. April 8th, we have James Moore and Denise Kirster speaking. April 15th, two speakers. April 22nd, Deborah Peters from USDA, who works on very large scale, multi-scale modeling of animal zoonotics. And we'll be talking about how to organize very large scale consortia. Then you see the rest. As always, we are delighted to have suggestions for future speakers. We remind you that prior speakers are welcome to speak again after an appropriate interval if you have new things to say. And so we hope that people will enjoy the lineup that we have for the next few months. It's a reminder for the rules of the meeting, and we always would appreciate in-meeting questions. I didn't clear with David and Jacob whether you're available for the half hour after the meeting to discuss in more detail. If you are, we have the breakout rooms set up. If those breakout rooms work, wonderful. If nobody shows up to your breakout room, then you're free to go. Last week, we had a lot of breakout room participation, and that was a big success. I hope that that will be something that people will use. And I'll especially today, because we have a slightly longer agenda than usual, I will provide a 5-minute warning, 10 to 15 minutes into the talk, so that our speakers are keep on schedule. We need to finish the talks definitively by the hour deadline. That's why we have the half hour afterwards for more free-form discussion. Hope that's okay for people. And I do appreciate that the short form is difficult. These are teaser talks. And with that, we'll turn it over to David. As you know, I don't do much of a preamble. I think almost everyone will know him already, and the exciting work that he's been doing as a teaser. And I'm sure that we have lots to ask him about afterwards.
Great, thanks, James, and thanks to the group for inviting me to speak today. And James, I'm afraid I do have office hours for my class right after this. So maybe if I can just put my email in the chat there. If people really want to follow up, I'd be happy to schedule a time to meet that works for both of us by Zoom or phone, however you want to do it. How does that sound? Okay.
Okay, that's fine. I'm sorry if we picked an inconvenient time for you, but that's, I appreciate you letting us know.
Sometimes nobody shows up, but can't be guaranteed. So I probably should hang out there. All right. So should I proceed then, James?
Please go ahead.
Yep. Okay. Right. So I'll tell you today about some of the work that we've been doing. Is that the correct or incorrect view? Is that like the presenter or the?
Yes, it's good.
It's good.
Yeah,
You don't
that's
see flag.
correct.
Okay. Thank you. Okay. So here we go. Yeah. So our normal research, let's say, has been in recent years in the cancer space and focusing on really the physics of migration. And we're trying to expand that to like the whole tumor simulation of cell division and death. That's been our main thing. We've been funded by the National Cancer Institute for the last 5 years with the Physical Sciences and Oncology Center, PSOC. And that work is multi-scale. We're trying to span from atoms up to tumors, and the time scales from sub-nanosecond out to weeks. And that's been our normal thing. I did want to tout the meeting that I did earlier, but I can skip that. But as you all know, and everyone had the same experience, a global experience of, we had to shut down our lab about a year ago. We do microscopy and imaging to test our models, and had to shift the lab basically to this place where I'm sitting right now. That's my cat Simone. And kind of started to think a little bit about, was there an opportunity in COVID? There certainly was a need to try to understand what drugs might be useful in the clinic to move forward into clinical trials that might be useful as antivirals, for example. And there literally were hundreds of proposed drugs and thousands of trials that opened up globally and are still open. And yet we don't really have a solid antiviral, certainly not one outside the clinic. And so this is still a pressing need. And that's what I'll tell you about today, is how about a year ago, I connected with two of the clinical trial PIs at the University of Minnesota who were running the hydroxychloroquine trial and the remdesivir trial, David Boulware and Susan Klein, respectively, part of, in Susan's case, a larger national trial. And what we endeavored to do was to build a model for the replication life cycle of SARS-CoV-2, basically leveraging extant knowledge on SARS-CoV-1 as our starting point under the hypothesis that SARS-CoV-2 would not be terribly different from it. Of course, we didn't know, but what else did we have at the time? So we started to build a model from that scaffold, from that literature, guided by experts in the field, so infectious disease and immunology experts highlighted on the right-hand side in blue, as well as the clinical trial PIs highlighted in green, and some of our PSOC investigators highlighted in gold. And that team then worked together over the course of about 2 months, meeting weekly and iteratively developing modeling, a model developed by Brian Castle, a postdoc in my group, a differential equation-based model, mass action, kinetics, and so forth, guided by the literature to produce this map on the left. And we want to map specific drugs onto it to see if those drugs were going to be targeting what we might call vulnerabilities in the replication life cycle, those parts that if you did target them would be predicted to have a big impact on the rate of replication as measured by the number of virions produced over time on the right-hand side. So if you could change the time that it takes to produce a burst of virions from a few hours to much longer than that, that would be a very sensitive part of this system. So we started to do that, but put that model together and identify where those sensitivities are. And we had to, see whether our model was decently predictive. We withheld some of the literature as used some for training and some for testing. So the grayed out ones are the ones that were used to test, and the black ones were used to build for training. And it actually hit a number of things that were kind of intriguing. For example, the ratio of genomic RNA to negative sense RNA was at least 10-to-1, and number 8 or letter H there, for example. So it actually captured some things that it wasn't obvious that the model would necessarily capture when we tested it after training it with A, B, C, and E. And then we looked systematically, single parameter at a time, then varying from that base parameter set to find sensitivities in it. Of course, some you change them, it doesn't really change the outcome at all. KXO, for example. Others are moderately sensitive, like the middle case, and then some are very sensitive. This RDRP step is a transcription, is the stepping of the transcription process. And if you go system-wide then across all the parameters, you can kind of see now a map of how, if you start with the base parameter set in the middle and make fold changes one at a time in them, up or down. And here we're thinking going up would be an inhibition of that step, each of which is required for the overall replication, which ones are most sensitive. And you can see as you go up, some of them you hit a very, a big effect, like a didn't even really complete the cycle on time scale of tens of hours in the case of this HRDRP parameter. Others, were not as sensitive, like this one, the second and third parameters that involve viral entry were not predicted to be very sensitive. And that's where hydroxychloroquine, for example, would be expected to map, whereas remdesivir would block the transcription more down the middle there where the green arrows pointing. That would be predicted to be a better vulnerability to target in the SARS-CoV-2 life cycle. And I'll tell you a little bit too today about fluvoxamine as another one is targeting ones that are associated with sigma 1 and 2 receptors. Further down, these are the first two of the three kind of vulnerabilities. And the third vulnerability is in translation, protein translation. And I'll tell you a little bit today about metformin as a way to target that and where we're at. Okay, so just to summarize then how that map looks, then there are certain nodes, certain parameters in the model that are predicted to be vulnerable and therefore effective if targeted, everything else being equal, and others that were predicted to be less effective at or able to be targeted. And I just want to say that, with a sufficiently strong inhibition, even some of these less effective targeting strategies could still work. So it doesn't mean they wouldn't work necessarily. It just means that everything else being equal, you'd rather go after the green parts of the diagram and not the red. That's where the sensitivities were predicted to lie. So that was where we ended up, and we put that on bioRxiv, and encourage you to check it out. And if you think there are collaborative opportunities based on that, I'm happy to kind of engage with you around that. But we started to then shift into how can we apply this or test it? And of course, literature was coming out, and really an important study, I think early on, was from a group that did a repurposing study of a lot of different drugs and looking at the protein interaction map to look for potential targets and drug repurposing, and which parts of this process were popping up in their screens as the ones that were most vulnerable. And it did align with two of the three hotspots that we identified, specifically translation and sigma-1, sigma-2 receptors, that third vulnerability in a later stage of the replication downstream. So that was encouraging, but we want to see, could we identify a drug or drugs that would hit those nodes? Like, can we use this as a map to guide our own clinical trial development? And this is where I started to work with Chris Tignanelli, who was one of the PIs on the COVID trials here, to identify, and here using his skills in natural language processing and informatics, to identify specific drugs that based on that method would be predicted to be effective. And a good alignment came with metformin, which can inhibit mTORC1 and among other things, and in turn retard the translation of the viral structural proteins. And so we started to look at metformin as a potential drug, and some others too, but this one really stood out. It was corroborated by some initial human lung tissue data that Matt Schaller and Greg Sawyer obtained at the University of Florida. And you can see, for example, metformin, 2 of the 5 donor patients having a pretty good response to it. It's interesting, that heterogeneity, like that, this may be the case that some patients are going to respond well for reasons we don't fully understand, but maybe encoded in the map that I showed you, and others may not. And similarly, sirolimus, which is another translation inhibitor, also had a pretty good effect. So the reasons why we want to move forward with metformin clinically would be that our biophysical model predicts that this is hitting a good target. Chris's natural language processing identified protein synthesis and specifically metformin as a good candidate. The drug repurposing study by Gordon et al. In Nature identified metformin as it's one of the few drugs that they could show in vitro had a decent effect. And Chris and coworkers did an observational study here in the Twin Cities on people who are already on metformin, and then their response to SARS-CoV-2 challenge being better than everyone else, but that's observational, so it still would require a prospective randomized clinical trial. And then the in vitro study that I showed you with human lung tissue. And lastly, and this is pretty important, is ideally this drug is already out there, it's available, it's relatively safe and inexpensive and available globally. And I think this is important if we're going to have global impact with these, and not a very expensive drug that's only available to a small fraction of the world's population. So this really hit a lot of the marks. And just briefly, here's the observational result. It's not real strong effect, but it's statistically significant, and it suggests that metformin is worth further consideration, at least. So all that packaged together, what I just showed you on that the list, is led to a COVID trial that's enrolling now for metformin. We've enrolled over 30 patients. We have sites open now in the Twin Cities and Chicago, with one in Los Angeles and Denver that will start enrolling soon. So that's where we're at with that. Within 12 months, less than, starting from scratch and having a phase 3 clinical trial that's currently enrolling. And we hope to complete that enrollment. If you have ways to, if you're interested in that, being a part of that trial, if there are any clinical trialists in the group here, be happy to chat with you about it. I'll just point out a couple other directions about the trial is that we're looking at expansion because there are 3 vulnerabilities, not just one. Protein translation is one, and we think metformin can have some impact there. But the sigma-1, sigma-2 receptors I mentioned as well led to identification of fluvoxamine as potentially effective. And so that's another one that we predict would be a good one to focus on. And we're going to likely going to expand our trial to include combination between metformin and fluvoxamine, as well as a possible third agent. So a multi-arm trial, multi-site, randomized, placebo-controlled clinical trial. And John, I got just like 1 or 2 more slides, and then I'm happy to take questions. How's that sound?
That's fine. You're right on target. So, and we'll
Okay.
do
Yeah,
your questions now, because that if you can't stay later, it's important that we have the opportunity.
Oh, okay. Yeah, that sounds great. Here's our current scorecard. And remdesivir at the top was one of the first that was given emergency use authorization. Certainly not a home run, but it has some utility in the clinic and is in standard practice right now. You go down the list, hydroxychloroquine and lopinavir, ritonavir were pretty clear failures and multiple attempts. So, and Dave Bower, who we've been collaborating with, was the PI in 2 of those hydroxychloroquine trials. So we shared our results, our prediction rather, with Dave before we knew the results of his trial. And he wouldn't tell us the results of his trial before we told him what our prediction was going to be. You go down the list, there are various stages, going from less certainty to going to lower certainty at the bottom. And I mentioned fluvoxamine already. I think that's already had a efficacy signal in phase 2. So we count it as, at this point, provisionally correct. So that's kind of where we're at. It's 7 for 7 in predicting efficacy in clinical trials so far. We'll see where that ends up, of course. We've got some preliminary human lung tissue data with Matt Schaller at Florida to support that. And I think the big picture is I'm becoming more convinced we can use biophysical modeling to objectively prioritize which drugs should be moving forward in a rapidly evolving situation like this. And I'm especially heartened, even in the face of variants emerging now, as we might expect, that the model was built on SARS-CoV-1 and seems to be predictive of SARS-CoV-2, suggesting robustness with respect to variation. So I encourage you to look at the preprint if you're interested, and I'm happy to answer any questions.
Okay. We were talking the other day about wanting to have success stories about modeling going to clinically meaningful results. And here we have one, and a quick one. So I hope there'll be some time. We'll reserve a little time for some questions, since the discussion for Jacob can run over a bit. Any questions for David while he's here?
This is John. I do.
Please go ahead.
Have you considered the possibility of going either to the Department of Defense or the VA, who has thousands of retired patients who are on metformin, and they know who they are, and taking a large sample of them with a very simple survey, 2-question to see who thinks they had COVID and who didn't, compared to a sample of the population in general, just to see if, in fact, you had a lower percentage of incidence among people who were taking metformin for its primary purpose.
I had not considered that. The retrospective observational study I mentioned was done by Chris Tignanelli here at University of Minnesota using a pretty large data set that United Health Group has here. That's a good suggestion, John. I'll pose that to Chris Tignanelli and see what he says and let you know. How about that?
I'm just curious and kind of naive. I take metformin and I did not have any signs of COVID. So, and a 1.
That proves it, John. I'm glad that it turned out that way.
Time for
I am too.
one more question, perhaps. 1 or 2, anyone?
So what is a working range for the dose of metformin that model predict?
Yeah, we looked at what's clinically achievable, and I think like what's typically used around 10 micromolar, 20 micromolar, something like that. That's kind of, a lot of people are using metformin for managing pre-diabetes, diabetes, and some people use it for weight loss, and some people think it's even an anti-aging drug. So that's the clinically observed range, and that's kind of what we're that should have a significant effect.
Other questions? We certainly have time for one more question.
Can you explain in simple terms what does it do without getting into details? Like what does metformin do that might target in simple words?
Yeah, it binds to there's this mTORC1. That's the central hub protein that regulates a lot of cellular processes, but one of the major ones is protein synthesis. So in simple terms, the virion, the virus, to replicate itself has to make lots of copies of the proteins to assemble into new virions. And that is a especially sensitive part of the cycle. Every part is required, but targeting them targeting the same way doesn't lead to the same output. Some are very vulnerable and some are not. That's a vulnerable one, we predict. Yeah.
I guess I have one question which may not be short, in which case we'll have to move on, which is the usual problem with antivirals is that by the time you have symptoms, it's too late. Now, in the case of COVID, where you have where you potentially have the COVID moving from the primary source of infection to secondary organs, then antiviral inhibition may be very important. Do you have any insights that you could share into the kind of issue about whether there are particular blocking strategies that might be more effective when used for light dosing antivirals as opposed to sort of prophylactic classic antivirals? Because remdesivir seems to really work as a prophylactic better than as a after symptoms drug.
Yeah, it might be maybe a reason why remdesivir is not strongly effective or stronger in its effect is that it's entering late in the game, and it's kind of not the key issue that needs to be managed for those patients at that point. So the earlier we can get these into the patients, the better. The Metformin Trial and fluvoxamine are both outpatient. So as soon as someone tests positive, they can fill out the survey to potentially enroll. And then it can be, currently it's local courier delivery from our sites in Minneapolis, Chicago, and we will expand to Denver and LA. But we're going to try to amend the IRB so that we can ship by FedEx anywhere in the US so that anyone can enroll. That will create a little bit of a delay compared to courier, but not much, maybe like half a day to a day delay at most. So that's, we think, still early enough. And the clinical endpoint, primary one, is hospitalization.
Very well. In your case, because metformin is so widely used, the prospective trial, the retrospective trial may actually be the most valuable, because if you find that it's protective in the people who've been taking it already, that's really, you have the opportunity to actually do a prophylaxis study, which is usually very hard to do.
Yeah, and I guess as a side comment about the observational results, they're kind of maybe more striking than at first glance, because you're talking about people who would be pre-diabetic, diabetic, which is considerable vulnerability for COVID, and yet that group had lower incidence of hospitalization than the control, were those not on metformin. So there's obviously demographic differences, but that's one that I think is kind of intriguing.
Yeah, I apologize. There are so many fascinating questions coming off your talk, but we've got to give Jacob his 20 minutes. So thank you so much for being willing to come. I hope you'll come back and update us on this. And,
yeah, thanks a lot
really appreciate your participation.
Appreciate you inviting me, and I appreciate the time. Thanks, everybody.
Okay, so we'll move on to Jacob's presentation. Jacob's generously agreed to be the second victim in the presentation of the subgroup results. And I'll turn it over now to Jacob.
Okay, I'll try to share my screen. Hopefully it will work. Here we go. Okay, before that, I plugged in some links in the chat. Those are links to our working groups. What I'll be presenting is a white paper that was composed by 2 working groups together. The first one is the model of reproducibility, credibility, and standardization subgroup. I'm talking about subgroups, not working groups. It's all viral pandemics working group. You can see Sharif here is my co-lead here, and he'll briefly come in and talk about some things related to reproducibility. However, in my sins, I signed up to lead more than one working group. So the other working group is the integration working group. We have multiple co-leads there. We have Wolf Bodness, Yaling Liu, and Robin Thompson. So, and this group was actually integrated from several other working group subgroups that were created at the beginning. We're trying to channel as much as possible together. So you'll find integration within host and population scales in this working group, the subgroup integration within and across scales, and ensemble modeling. All of those are integration processes that we cover in this subgroup. So if you're interested, we created those SimTK sites. If you're interested in any of those topics, please sign up. We have a mailing list. You can join through here. If you press, you'll have the ability to subscribe here. We started the activity I'm showing a few months ago, around December, and I'm showing some of our activities. You can actually find out what we're doing in December, mostly integration. And I saw Filippo Castiglioni here online, so he was active there. Later on, we talked about some other issues, and now mostly we're talking about the white paper. The last link I sent you is for the white paper itself. White paper is accessible online. I will bring it in for a second. This is the white paper, and it's all done as a Google Docs. And as you can see, it's quite long with many, many references. It was composed by contribution by many people, and I'm going to talk about it later. It started as 2 different papers. Each working group, each subgroup had its own paper, but then we figured out there's a lot of commonality, and we just decided to integrate them. And what you're seeing now is the fully integrated paper. The integrated version I took, and I did the editing. So basically I'm the one who's editing here, but I made sure that I take all the references from the original 2 papers, and each one of the contributors, you'll see in a second, contributed text there. I tried not to delete anything unless absolutely necessary, just to make things possible and readable. So anyone who contributed even a few lines or a few references supposed to be represented here to some degree. And I summarized everything, and I'm going to talk about this briefly. I also, as you can see, we have many, many contributors, 17 of them. We had one more contributor who decided not to be affiliated. You can see the discussion publicly on the mailing list. so I'm not sure if I should say his name or not, because he actually contributed quite a bit about annotations, but he's not on here on the list. There is one more contributor who was anonymous, somehow slipped in at some version, added the line, and we couldn't locate who the person was. So since it was one line that is not in the paper, the person is not here. Otherwise, you will find all of the names here contributing, and you can actually trace back exactly what they contributed, because Google Docs does support versioning, and you can actually see who contributed what. So I presented the working group, the subgroups. I presented the contributing author. One more thing, Sharif has still not approved the paper. Sharif, hopefully you'll approve it. All of the other contributors have approved the version I'm showing you now, except from Sharif, but he'll jump in and he'll say what he wants about this paper later on, because he has a part here that he will speak about. I looked at the chat and I saw some of the contributors are here, so maybe I'll try to redirect some of them so they will explain what they contributed. But I asked it to be 1 minute. So the introduction itself was written mostly by, actually not mostly, but Eric Ferguson started the introduction. He explained why models are important and why the old SIR model is becoming obsolete and why there's a need for better models. But then James Glazier and several others added. And Winston Guerrera, I saw him also here on the chat. He contributed something in the introduction. Later, some of this text was moved to a different section, but Winston, you want to say something about what you contributed? He's not jumping in, so maybe maybe he'll stay to the room for later on. The next thing we were discussing about credibility of models, and John Rice, he actually was contributed quite a bit about that. So John, if you jump in for a minute and say something about what you were talking about, because it led us to many things.
Yeah, my input pretty much was around the concept of model credibility. And that's my background for a lot of years in defense before I retired 12 years ago and got involved in the stuff you guys are doing. But my input was from the perspective of a co-chair for the dissemination working group with the very simple thesis that effort to disseminate models that cannot be shown to be creditable is probably more damaging to the modeling and simulation effort than it does good. And two of the key things going on there right now is that on the industrial side, commercial side of modeling, where more medical industry is dealing with regulators, there has been huge movement toward the development of standards specifically associated with the accreditation of a model. And so we have the FDA who says, if you want to submit data for clinical trial consideration that came from a model, we, the FDA, have to be the accreditor. And so the added theme to this paper was modelers are not the accreditors. Those of you who build models do not accredit your own models. Your only responsibility is to provide all the detailed information that somebody who wants to use the model has to have so that they can accredit it. And then the responsibility for the results of using a model rests on the user, not the modeler, because it was the user who decided that that model was appropriate for their use. So the utilization theme and that kind of stuff leading to credibility and where the standards for documentation are going was my input.
Yeah, and this led us to something that Sharif actually noticed when John was talking about this. So Sharif, jump in and please say and explain the diagram and explain your part here. It's the next section.
So I was I added about this reproducibility in the manuscript because that's the starting point. If you cannot reproduce the model, then there is no question of credibility or using the model or integrating the model. So that's the core issue and the first issue. So many of you might have attended my talk where I showed that the results of our analysis where we analyzed about 455 models published in several peer-reviewed journals and found that about half of them could not be reproduced based on the information provided in the manuscript. And we found several issues that many informations were missing or incorrect. So you cannot reproduce. So many do not share the codes, or they share wrong codes or provide wrong information. So all these different small aspects are very essential for reproducibility of the model. I think in the later part of the manuscript, we discuss in details each and every aspect. When we have models in different codes, it's going to be difficult to put them together and reproduce them. So I think I will leave Jacob to speak more on that. But in my case, in my section, I was focusing on the reproducibility aspect. And there are many other general aspects which are touching upon the reproducibility. So here we started with this reproducibility. First, the model should be reproducible, and then it should be credible enough. It should have enough information that we could use it, and it should tell the purpose of the model so that we can utilize it properly. And then it comes to integration, where we can integrate multiple models and get the broader picture. I think I will let Jacob take on from here and explain further.
Yeah, so the diagram that Sharif created here actually explains well why the subgroup for credibility, reproducibility, and standardization is related to the integration one. We add a lot of topics together because you cannot integrate unless you have all of the pipeline of things that build the model, meaning it's reproducible, we believe it, it's credible, and we can use it. Only then you can integrate. So this led us to many, many topics by many contributors. We don't have enough time to speak about it, and the entire paper is 20-something pages long. So I'm going to try to give some glimpses and briefs of what we did, but this table summarizes what we came up with. This is the work that people did for several weeks. Integrated as best as possible. You can integrate work by so many people. So we have difficulties on one side, and we have expected or proposed solution on the other side. The solutions are not 100%, but those are like glimpses of things that will maybe evolve in the future. But we do need to solve those difficulties. Most of them we have solutions to some degree, some we don't. So we talked about credibility already. I will skip it. Basically, we want better practices, the solution, and documentations and tests. However, we have a problem with model validation that the team didn't come up with solutions. And the model validation comes from two different aspects. One of them is multi-scale models. You don't have necessarily data from all scales. So this is why it's difficult. More than this, if we're talking about forecast models, well, you cannot really predict the future. How can you get the information to the future to validate against? So basically, those are two difficulties discussed. We didn't have any solutions there, although there are some existing ones in the literature to some degree. We focused on other problems that we see, and there are many more other difficulties in the way of modeling. One of the first ones is models are written in different languages, and you can see actually in the mailing list of us communicating and trying to convert a model. I think it's Robin Thompson and William Hart had a model, and I tried to integrate it, but they wrote in MATLAB, I was in Python. Oh, the data doesn't transfer. Oh, we have to do something else. Those are difficulties that exist all the time. However, good thing is that the other people thought about this problem. There's a whole community with the system biology markup language, and which is later joins the community of something called Combine, which combines many communities that are trying to solve those problems. System biology markup language, or short SBML, provides a solution which is a language that allows you to represent models that can be implemented in later in other systems, other tools. It's not 100% a standard. It's something that they are trying to standardize. There's still a lot of work in this specification to do to make it a standard, but it is one of the best solutions that are available today towards that. Also, we want people to better document and annotate whatever they're doing. Another problem we discussed is even if you can.
I just need to warn you that at 4 p.m. your talk officially ends and you have less than 7 minutes.
Okay, I'll run it through. So I'll just almost read this. Models are hard to locate. There are several solutions, and you can see here listed a few repositories. Biomodels is one of them, CMCK. IMAG-Wiki actually have some, and there's a new one called Model Exchange. People have to know about those existing. We have problems with platforms that allow you to execute models, and this is also to join the dissemination working group, because if you can execute models on platforms that people can use and collaborate online and see the results of the models, it will help us. There's some of them that appear, biosimulators and non-biosimulations. We have models that themselves require adaptation. For example, we were integrating multiple infectious models. Some of them were expressed as density functions, while others were expressed as relative to maximum infectiousness. So you have to calibrate there. Also, there's an issue of scales, and scales have to do with units. We took a mortality model by Filippo. He's here. His scale was time step is 8 hours, while I needed 1 day. How do you integrate this? Well, this is simple. It's translation of units, which is our next section. However, units are not standardized, and translation of units is really a mess. If you go to clinicaltrials.gov, you'll find that there's like tens of thousands of units available. And I have to declare a conflict of interest there. I have a solution called clinicalunitmapping.com. Which is supposed to integrate multiple standards using some machine learning. This is one solution, but in the future there might be others, because there are multiple standards, IEEE, UCUM, CDISC. There exist, even units are not have multiple standards to use for. So it's amazing that we are here in 2021 and still don't have solutions for this, but we might in the future. Data measurement also and definitions are a problem. Even diseases in clinicaltrials.gov, when you go and look at the definitions of outcomes, they differ. But within the models, how do you decide where a model does something? Like we have Castiglione mortality model. Where does he decide to put the cutoff to decide the organism dies because enough cells died? Those definitions are important. So it requires some solutions like human interpretation or new measurement devices. And actually unitization, if I see Hanna Dubowolski is here, so she contributed something there about new devices not having to measure the data differently about how much virus it is. Hanna, you want to join in? She's not responding, so I'll just skip it. We have problem with annotations. There's an organization called Combine that has some policies about it. Read this in the paper. We also have several other problems like implementation barriers. The models we see today are the ones that pass the implementation barriers. We still have many models that are out there that people have to implement, and we have to solve those. So the solution there is better education. And again, I'm skipping many things. The paper has many more other aspects. There's a licensing problems all across the way. Open source licenses, even if the word sounds nice, they're actually incorrect. Most of them are copyright based, meaning, and copyright is a legal restriction tool. So you cannot always use it. Oh, we're done?
We're going to have to why don't you leave your slide up and
okay.
Let me simply call for other business, and then we'll have time. You'll have your half hour to discuss these issues.
Okay.
But I have to formally close the meeting.
That's okay.
So just to come back for a second, and then we'll return to Jacob's work. I just want to say thank you to our speakers. Thank you to Jacob for presenting that material in a very impressively compact way. And again, we have time now to talk about it in our informal discussion. I won't be using the breakout room since we only have one person. We'll leave it in the main session, so there's no need to join a breakout room. We would love your suggestions. Definitely suggestions for publicity. As you know, we had a slightly smaller turnout today than usual, despite having two very good presentations. And so your ideas about ways to improve our PR would be most welcome. We are working on a leadership statement and a goal statement, Reinhardt and I, and we'd like your suggestions for that. And so with that, I would like to close the meeting unless there are specific suggestions. Is there any other business that people would like before we move on to the open discussion with Jacob? No, in that case, I appreciate it. I'm sorry, Jacob, but we have to formally close the meeting for 4 o'clock. With that, I'd like to thank everybody for participating. I will return the floor to Jacob for the discussion session, and that will stay in the main room, and we can continue till 4:30. So Jacob, back to you.
Okay, so I'll share my screen again, but you have to give me the permission. Here we go. So back here, basically the rest of the topics are something that we can talk about it, but we have identified many topics and eventually we had many open issues. So we plan to publish this paper soon. We have permission to publish by most of the authors. Some of them are here. Sharif is here. I still want to see why he hasn't approved yet, but because we cannot publish unless we have everyone approved. But now it's open discussion if anyone is interested in commenting, saying something, contributing, joining us. Everyone's quiet, probably. Yeah, probably 4 o'clock was a cutoff time for them.
No, but it's fine. I mean, Rahman is on, John is on.
Yeah,
so
I see Winston is here as well. He also contributed.
So I would
yes,
like
I did contribute. Perhaps I just add a few remarks on model integration. That I said that there are two issues on a model integration, which is the biological integration and the mathematical integration. And the biological integration is normally achieved through exchange of organisms which are involved in the disease dynamics, that is the pathogen and the host. So that's where the biological integration happens. But after that, then there's need to convert that into mathematical integration. And because normally when you have scales, when you are doing multi-scale modeling, the different scales actually indicate changes in processes of the exchange of these organisms in disease dynamics. So you have to upscale and downscale, which is the mathematical difficulties that we face even up to now when it comes to model integration. And that's the major challenge in model integration. There are few successes that have happened. There are a few papers that we have written, even myself and a few others who have contributed to model integration. So I think, but that's the most difficult part, which is converting the biological integration into mathematical integration of the submodels. Thank you.
Yeah. So this is one approach that was discussed. there was other discussion about model composability or how will you pronounce it? Composability is the correct pronunciation? Composability, yeah. Yeah, okay,
Yes.
Raman says yes, I pronounce it correctly. So model composability apparently has mathematically been defined. We have William actually contributed this information, and James, you liked it as well, I remember. So you thought that it has to be expanded. So we'll have to expand it at some point, but this is something that we discussed and we'll have to address more later. And this is where it leads to what Winston was saying. It's like this is the mathematical part he's talking about. But we need to actually elaborate on it and make it clearer. And apparently, model composability has several issues. One of them is serial, like this leads to that. The other one is things that happen in parallel. But it also, there's a level of the ensemble. What are things that are assumptions that happen in parallel? This is a different level of composability that has to be looked at. So thank you, Winston.
Okay.
James, you had a lot to say about the paper at some point, and please, now is your opportunity.
Well, I think actually something that Winston said I'd come back to, and it's there in the paper up to a point, that I think that it's important to recognize that there are at least 3 different levels of abstraction of these models. I promised you, I think, a picture, and then I didn't give it to you, but I can give it to you. Which is that there's what is formally called a conceptual model, which is what a biologist uses, which is a list, a verbal description of what's happening, with no mathematics. It's a list of what the objects are in the model, what the processes are in the model, what the interactions are, what the dynamics are, what the initial conditions are. But those are words, maybe words and pictures. There's a second layer, which is a mathematical or quantitative model, which says that you're going to make certain assumptions about how those qualitative objects that are described in the conceptual model behave. Physical object obeys Newton's law. Physical object is overdamped. Physical object diffuses. Those are mathematical. And those are intrinsically less shareable and less general than the underlying reality, which is described at least more completely by the conceptual model. And then below that, you have the scale of specific computational instantiations. Do I use a particular integrator? Do I solve it stochastically, or do I solve it deterministically? And those are much less shareable again. It's not that you can't do sharing at each level, but the knowledge that is encapsulated in the model is primarily available and shareable at the level of the conceptual model. And each time you go down a scale, you lose information. You gain certain kinds of information, but you lose information as well. And so one of the things that makes model reproducibility so difficult is that most biological modeling papers do not present a formal conceptual model. And I could, again, it's easy enough to show how this works. SBML was to a large extent designed to allow you, for at least chemical reactions, to present both conceptual models and mathematical and computational models using the same framework. So the basic concept in SBML is a chemical reaction. You say A goes to B. 7A plus 9D goes to 4C plus 8D. So you actually write things in the forms of chemical reactions. That doesn't necessarily have any mathematical instantiation at all, because if you don't don't say after that what the rate is, what you've defined is a network of reactions, but not a mathematical quantitative model. And again, those SBML models, once written down, can be implemented using Odde solvers, Gillespie, stochastic solvers, and so on. So SBML worked very hard to try to keep the conceptual models integrated with the mathematical and the computational models. For multicellular models and other kinds of scales, in general, we don't have any formalism to do that because there's no agreement on how to describe, say, what a cell does. And so there's a problem. But even then, one can still present, if in the form of a table, for example, a formal conceptual model. And one of the reasons I specifically wanted to invite Deborah Peters to speak is because this is her area of expertise. How do you build conceptual models of very, very complicated processes? And she needs to do this because to reach out to ranchers, to meteorologists, to geologists, to people studying insect behavior, to people studying economics, who all have to understand the model, it's critical that she has formal descriptions of the conceptual model. These are what an engineering company might call product descriptions. And we tend to short circuit that because they're time consuming and verbose when we're doing research. But fundamentally, if you don't go through that level of description, you're building models that are thrown away. And I think it's really critical that that people understand that current modeling practice, for the most part, doesn't follow best practice because they don't build those formal conceptual models. And I think I've said this before, but this goes back, it's not a new idea. In fact, this idea was enunciated beautifully and very clearly and completely in the Diderot d'Alembert Encyclopedia of 1780. So it takes some time, just the way for ontologies, Aristotle is still the go-to reference. 1780 is when this idea of the necessity of conceptual models was first really clearly enunciated. And it's interesting that we're still not doing it in a consistent way. So that's my speech. I've read it into the record.
The limitations that we face because of having forgotten history and ignored philosophical discussion, and very early on when I joined the working group, like day one of my first questions to Reinhardt, whose academic background was science philosophy, was like, whoa, are we going to talk about philosophy? And the answer is no, we really don't have time for that. And every time there's a discussion, it comes back to, well, we don't have a taxonomy of all the different kinds of models that's well enough agreed to that you're able to talk about conceptual models versus math. What is the difference? Where are the crossover points? And we just don't have that that I'm aware of, or we don't want to.
This is incorrect. I can point you to a paper that was written a long time ago about taxonomy of disease models specifically. And this is like 15 years old now, by now. So there is existing taxonomy, at least for disease models. I can show you the paper. I have to dig it up, but it exists. So it's
There's
just
no
that people don't read
funding. It might be fun, but it's not sexy to do. And so skip it and go do what they're funding for.
People did this already. John, they did this. I'll send you the paper. I'll find it and I'll send you the paper. Taxonomy for disease models exists, at least to some degree for 15 years ago. They don't have maybe the new advances. But if you're talking about new advances, James,
no, I know that stuff's out there.
So, James, you mentioned something about stochastic models. This is the last point in the paper, but do you know that the new models like the machine learning models are not reproducible because they run on GPUs? Did you know that?
Again, one of the things you have to be careful about also is is the term reproducibility, and reproducibility versus replicability.
Repeatability, they're not even repeatable.
Well, that goes back a long way. I remember when I was a graduate student and the original Sun 2, not Spark even, but the Sun 2, they came out with a floating point accelerator. that had a random error in the last bit. And so when you took sine of x, it had a small rounding error, which was different every time you took sine of x. We were doing models of chaotic systems where, of course, the error is amplified exponentially. And so every time you ran a deterministic model, you got a different answer.
Good. So the same
So
problem
this is
exists
this is
now.
an experience I'm familiar with. But a well-designed model, of course, should be able to handle that.
No. This is the problem I'm telling you. It's not a well-designed. It's the fact that the hardware will not support it. The reason is because you run things in parallel on GPU. And there's a race condition there that they haven't told it. So the algorithms that talk to the GPU, the basic functions are not repeatable. And this is an issue. I raised it, and they're not going to fix it in the near future. It depends on some algorithms.
This is a good reason to explain why computational instantiations of models are not repeatable and shareable.
No, this is specifically for the new machine learning stuff. I'm not even talking about the biological. It's because the subroutines that talk to the GPUs. If you run it on GPU, no, it won't be repeatable.
But again,
you know, Louis.
Anyway,
Louis
no, I don't want to
Lorenzo,
waste time on this.
Louis, Lorenzo, and Giuliano are hanging in here in this discussion.
They haven't said anything.
And I don't hear them. Hello, guys.
They may be doing other things too.
I was just enjoying the discussion, to be fair.
Welcome.
Me too.
Louis, what do you do?
But I think these are all issues that are important. But again, you want to be a little careful. And what you're talking about, Jacob, is replicability rather than reproducibility.
Repeatability even. You run the same thing on the same machine using the same software, it will not repeat.
Right. That's replicability, technically.
I know that there's repeatability because it's like you cannot repeat the same thing. And those definitions change each time you go to a different form, by the way.
But I think it's certainly very significant. Of course, what you mean by the same is an interesting question. If you believe that your model is deterministic and it isn't, that's serious.
Yeah.
If you don't care about the stochasticity, or you accept the stochasticity as part of the model, then what you're saying is that those models intrinsically have a stochastic element, but they're advertised as deterministic.
They are not advertised as deterministic. The machine learning people say it's okay because if it's fast enough, it's good for us. This is the argument. We had this big argument in the Python meetup about that. We actually have an issue on GitHub in one of the libraries. It's like it's an NVIDIA library, Rapids AI or something. Like we discussed it to the point where we understand that there's a problem there. They don't care. They're not going to fix it because it's good enough. It sells.
Oh, but good enough may be, but that's exactly the point. Is that good enough? When you define reproducibility or replicability, you typically define some kind of tolerance, which says that you accept the result as long as you're within some degree of difference from the original result. It's legitimate to say that it should be bitwise identical, and that is one standard. Another standard is that you're within some tolerance.
You're thinking about mathematically. Those are machine learning. The new machine learning models don't care about this the way you think about it. They're not thinking scientifically. They're saying, oh, it explains it can translate the language, the text, it's good enough for me, even if it's like 90 or 80% of the time. If a human accepts it, that they're good enough. And usually they can show you lots of graphs, how good or how bad it is. And they can claim on this data, on that data, it works, and on the test, on the and on the training set, this is about accuracy. And this makes sense. But those tolerances are actually something that's like, it's the best we can do. And they say if we have faster processors that get to the same way, same results faster, we don't care if we can reproduce it or not. If we got one model running okay, we're good enough with that.
So sometimes you have problems where you try to match what the computer predicts, what the computer tells, and the computer tends to be much more reproducible than a human, and what you and your colleagues tells. For example, I use this machine learning to basically segment image or to classify classes of images. And at the end of the day, when I test against myself and my student, I find that the difference between me, my best student, and a newbie in the lab is so wide that any error the machine learning does is better. And the machine learning will do always the same mistake. While if I have new student in the lab, they will do a new mistake, which is completely unpredictable. So in terms of standardization, I think the machine learning is helping, but I'm talking about image analysis purely. I don't know about other stuff.
I more than agree with the machine learning tools are good, and the argument that people make are okay, but I'm trying to tell you that it will eventually affect down the line the scientists who are trying to get to prove that whatever they're doing is reproducible or not. So eventually the stochastic modeling difficulties will remain for a long time with us.
Yeah, that was the other thing I wanted to ask. That is very interesting because on stochastic modeling, well, it's interesting because many of the biological process is ultimately stochastic. So, and we cannot do anything. If we try to make deterministic prediction, well, we will invent a reality that doesn't exist probably. And the problem is that probably we have already problems with computability. So how many calculation can we do per second now? And how this will evolve in the future, etc. And then actually implementing this stochastic stuff, then that goes beyond my expertise by far. So
you're talking sense. This is actually something that we're seeing now already. Stochastic simulation are incorrect, but also here, COVID, in each state or each country, it behaves differently, partially because of stochastic things. How do you model something like this at all?
Yeah, or things that you think are stochastic, but you simply don't know what is the rule underneath, and so for you are stochastic, and so you try to model them.
Yeah, I agree, but the thing is, it's hard. And so
Yeah.
you use stochastic methods to do this, and this is actually what Eric Ferguson was, his argument was. It's like, how does an SIR model that is deterministic supposed to solve something is stochastic behavior?
Oh, that's that's
Yeah.
a good question. So shall we give up?
No, he actually has solutions. His research is into stochastic is into how do we do this stochastically. It's actually in the paper, in the white paper.
Yeah. No, that's good. I really think I already, in my really state as a newbie, I already hit some bit where I say, that's not really deterministic, and I'm just guessing here. So, well, it's the two different cases. I'm just guessing here, and second, I'm trying to be deterministic when there is no way this will be deterministic in the real life.
Well,
So,
there's nothing wrong with stochasticity. And I think I would encourage you, it's an interesting thing to discuss, and there are real problems with interpreting stochastic models. But I also wouldn't get hung up on that as the be all and end all determinant of whether models are reproducible or not. No. Because there are many aspects of reproducibility and validation. I mean, in principle, if a model is well behaved and you run it enough times and it's stochastic, then you reproduce the distribution of results. Now, exactly what that distribution means in some cases is tricky. If your model produces spatial patterns, we don't have ways of comparing spatial patterns. And therefore, you don't have a metric that you could use. If your model produces a single output, like the number of people who died in an epidemic, and you run 1,000 replicas, and each replica gives you a different answer, then you can have a distribution. You can do the standard statistical analysis of those distributions. And so the issue is that stochasticity gives you certain certain things you can't inquire of a stochastic model. There's certain things you can. But you do want to know whether the model is stochastic or not. I think what Jacob was pointing out was that there are models that don't seem to be stochastic that are because the hardware is stochastic.
Oh, yeah.
This is something that came up many years ago when people actually developed true random number generators.
If you're talking about the random generators you're talking about is a problem on its own. I'll send you a paper from the guys who are doing the census. They needed it. They needed a random generator and couldn't find one.
So there were construction of hardware random number generators, and the difficulty with hardware random number generators is that you cannot reproduce the sequence of random numbers. But again, these are, in a sense, I think, technical issues that are very interesting. And certainly need to be mentioned, but aren't necessarily the most serious problems to reproducibility. The fact that one thing that happens, for example, in reproducibility, which things like Miriam are trying to deal with, is that the way the data are processed to produce the figures in a paper isn't published. And so a paper should fundamentally provide all the raw data, even if it's in an Excel spreadsheet, and then the series of transformations that were used in whatever system was used to generate the figures. And that's something that isn't generally done. The problem with most of these issues of reproducibility is that they're very time consuming and expensive to do. They're a pain, and people aren't paid to do that. They're paid to do new science. But if you really believe in reproducibility, then you need to do those things. And so I think it's a tough issue.
So the problem is with reproducibility, especially stochastic system, is whether you know your system is actually behaving. You cannot test it really. So a stochastic system, how can you test something? Even the debugger will not let you debug it properly. If you understand, because each time you have something else, you cannot find some error. And this is a problem. You have to be very strict.
Again, the issue of stochasticity is certainly one that you have to then run replicas and look for distributions. Rare events are hard to trap. If your model gives a wrong result for some rare event, then you may not see it.
Yep.
Again, there are random number generators which are supposed to give numbers between 0 and 1, not including 0 and 1, which actually occasionally throw zeros and ones. Those kind of things happen. And so there are all, I mean, as you say, there's this whole hierarchy of confusion and complications. But I think the kinds of things that Herbert Sauros worked on for years, which is these issues like make the raw data behind a figure in a paper accessible, and the series of transformations used to generate the figure, those are extremely helpful. That's not addressing the model, but it's addressing how the data are presented so that one can at least know how the data were interpreted. Because if you don't have the mapping of whatever the model produced data to the published information is, you often can't back it out. And
traceability. You want to trace back things to the source. Yeah.
So there are so many important issues here, and of course each one of us has our own particularly hobby horses and focuses, because we come to these problems for our own backgrounds, and so the experiences that we've had and the frustrations that we've had will color what we think are important and what aren't. These are all important issues, or the saliency of them will differ depending on the individual. And I'm